Good morning, everyone. This is Beth Galella from Bristol Myers Squibb, co-leader of the Pediatric Working Group, and I have the pleasure of introducing our speaker today, Karen Thompson. Karen is a senior principal scientist of preclinical development at Merck Research Laboratories in West Point, Pennsylvania. She's a research formulator of solid oral dosage forms, and her focus includes pediatric and geriatric formulation development and formulation strategies. Besides having been an active member on the IQ Pediatric Working Group, Karen is a participant in the NIH Pediatric Formulation Initiative. She enjoys the excitement of formulation design and development, seeking to devise solutions to enhance patient adherence. At this time, I'll turn the presentation over to Karen.
Thank you very much. It's really quite a privilege for me to share some of these thoughts today about extemporaneous formulations. I've been involved in the development of these formulations, actually for label changes for products. And at the end of the talk, I'm going to propose a strategy that I think will allow extemporaneous formulations to help us to better design formulations for pediatric patients, meeting some of the new requirements that we have for pediatrics. So I'm going to start off this talk with some definitions. What is an extemporaneous formulation, both from the U.S. perspective as well as this perspective in Europe. Then I'm going to provide the backstory. That is, what you see in the actual label is the instructions that the pharmacist will use to make a formulation specific for a patient, and the instructions for how to do that. But there's a backstory, if you will, behind those instructions, and I'm going to talk about what the components of those are, because it may not be so straightforward. And then finally mention some of the utility for extemporaneous formulations, I think, in pediatric formulation development today, and I will focus on two products and some of the reformulation efforts that were needed to be done or proposed to be done so that we could meet requirements for the regulatory bodies from a health perspective. So extemporaneous formulations are actually defined by the USP as compounding. It's where it's a preparation, mixing, assembly, or altering packaging and labeling of a drug delivery device or device in accordance with a licensed practitioner's prescription. And the medication order is initiated as part of the interface between the practitioner, the patient, the pharmacist, and the compounder. So it's really part of that interface with patient-centric care, where pharmacists may compound specific products for their patients because of some issue the patient may have with the existing marketed formulation. And I will say that state boards oversee pharmacies and regulate the practice of pharmacy. So it's beyond, if you will, the purview of the FDA. There was some discussion about that, but I will just say that state boards, individual states within the U.S. regulate this practice. It can be further defined as preparation of dosage forms both for human and animal patients. And that reconstitution or manipulation of the product may require the addition of other ingredients beyond just the commercial product. And the other thing I will just mention is that the preparation of the drug or devices for prescriber's office use were permitted by federal and state law. So again, this is for the U.S., and actually it's from a textbook by Lloyd Allen, who has been a real driver in the quality of compounding practice within the U.S. So in Europe, compounding is still done. And I will say that just a little bit of a history, from the 1950s and earlier, compounding of medicines for patients was the norm. After 1960 or so, we were manufacturing dosage forms, and if you will, the practice of pharmacy did shift. But even still, I would say that about 10% of products are still compounded by pharmacists to meet the patient needs. So in Europe, there is a practice of pharmacy, And in that practice, there's sort of the magistral formulation where a medicinal product is prepared in a pharmacy in accordance with a prescription for an individual patient. There's also the official formula where the product is prepared in a pharmacy in accordance with the prescription and pharmacopoeia and is intended for use directly by the patients. So in one case, you're looking at manipulation of a marketed product. In another case, you actually have a formula based on bulk drug where you're actually making it according to the pharmacopoeia. And I will say that there is at the EU PFI meeting last week, there was some discussion about the European Committee on Pharmaceuticals and Pharmaceutical Care and the European Pharmaceutical Commission. They've launched an initiative to make available a formulary for extemporaneous formulations for pediatrics, compiled as monograms for their preparation. Now, this is a desire to standardize, if you will, these extemporaneous preparations throughout Europe. The important thing here is that, again, the compounding activity itself falls under national competency for individual countries. So in the U.S., it's certified and controlled by states, and in Europe, it's certified and controlled by the individual countries. So the backstory for an extemporaneous preparation. An extemporaneous formulations are prepared by trained and licensed pharmacies, and they're interfacing, again, with patients for patient care. The development of the backstory is all of the information that is utilized to provide guidance and supporting information for that recipe, if you will, for making that preparation. It includes all of the materials. They're explicitly specified, sources, excipients, the vehicles if you're using those, packaging and delivery devices. It also provides in the filing to regulatory bodies stability and storage information around that preparation, including shelf life, and it can provide also PK information, clinical experience for that formulation. And so it actually is quite expansive beyond just the recipe that appears in the label. And I will just discuss this with a couple of products that I was involved with where we developed extemporaneous formulations for their utilization. And I will say, in light of some of the recent discussions today, it almost represents an ultimate in patient-centric administration. It's that human interface between the patient and their physician and the pharmacist, and what the patient needs, and then the pharmacist makes the needs directly for that particular patient. So materials. So from a development perspective, an extemporaneous formulation begins with, if you will, an existing, validated, marketed drug product where it already has all of the regulatory controls in place. So my source of API in these particular cases are based on this existing marketed product. All of the excipients within it are clearly designated, and so my starting point, if you will, is a reproducible, validated process. That product contains excipients that are known, and they become part of the extemporaneous preparation by default. And what one does in the developing of the backstory based on these materials is if I'm adding additional excipients, I'm going to look at the regulatory grade of those excipients, and I'm also going to provide a justification of all the excipients that I'm using because they're all part and parcel for the formulation that's being administered to pediatric patients or other patients. And in effect, this is true for any pediatric development. It's really no different. And then we're also going to look at that preparation in terms of its physical and chemical stability, and also I'm going to seek to optimize its pKa in its development. So an example that I'll give where the marketed product provides some of the excipients, perhaps some that I would not choose to use per se because I had to make some changes in the extemporaneous formulation as a result due to the development. But for example, for the lisinopril product, I started with 20 milligram lisinopril tablets that were available in the marketplace. Those tablets actually contain calcium phosphate as part of the compressed tablet image. And calcium phosphate is great as a compression aid, But when I take those tablets and make a extemporaneous preparation out of them, that calcium phosphate has a buffering effect. And what I found in the course of the development that when I used certain compounding vehicles, I actually failed antimicrobial effectiveness testing at the end of shelf life, not at time zero, because of the buffering effect of the calcium phosphate. So part of my development is to look at all the materials and the potential impact that they could have on some of the important portions of my formulation. The other thing to consider with vehicles themselves, and in the case of these preparations, I used some existing vehicles on the market. I wanted to make sure that the suppliers of those vehicles maintain my supply, because those vehicles are part and parcel to availability of my formulation, and that the components that are used in those vehicles actually meet the standard requirements that we would use for excipients. And so we had to involve the manufacturer of these compounding vehicles in some quality reviews of their processes, as well as this thing, assuring the quality of the ingredients that they used in the compounding vehicle. The other thing that you want to make sure is that if the manufacturer of these vehicles down the road decides, oh, I'm going to make a change in this vehicle, I need to have time to assess the impact of that change on my extemporaneous formulation. So I mentioned this, and I'm going to bring it up again. Once you actually make the recipe and you've done this whole backstory around your development, there is a certain amount of diligence that is required to make sure that that formulation, as things evolve, still meets those requirements. And it does require some additional work to make sure that that is the case. The other thing that's outlined in the extemporaneous formulation development has to do with the containers, the closures, and the devices. When I make my extemporaneous preparation, if I have subjected my formulation to light stability concerns, I may have some restrictions on packaging that I could use. And part of my development is to identify that so that I can specify in the recipe the packages and the contact surfaces that can be used. So we do all of that testing in terms of the materials of construction, assessing the suppliers and their controls, and then relaying that information to the pharmacists in the instructions for the making of the extemporaneous preparation. An example that I'll give is for the losartan pediatric suspension that we developed. This suspension actually produces the losartan as a solid form. Part of it is to crystallize it out in a fairly small particle size so that we get taste masking. And as a consequence of that formulation, making that suspension, we did see issues with adherence of the precipitated particles to various contact surfaces. So explicitly in the instructions, it's required that you use amber PET bottles for that reason. With both HDPE bottles and glass, we did find issues with sticking, which resulted in lowering of the overall assay of the suspension. So you need to look at these controls to make sure that you understand for that extemporaneous preparation that in fact you are producing the proper concentration, because the dosing is according to volume. Likewise, for the delivery device, we did do studies to confirm that syringes of various manufacturers could be utilized for this. It's not surprising. This is usually done for dosing that is done through GI tubes and the like to make sure that there are no sticking issues, that there is compatibility with the delivery device. And those things are also conducted in the course of the study. So the other thing that's done for these extemporaneous preparations, and what makes them a little bit different from, if you will, products that perhaps may have a 2 or 3 year shelf life, as a solid dosage form, these extemporaneous preparations are designed to be prepared and given to the patient for a finite period of time that's clearly defined. The stability of these formulations is not years. It's more on the order of weeks or months, depending on the dosing period for the patient. In the case of the development that we had done, we actually had followed the stability over a 6-week period to support a 4-week storage label. And in the course of testing of that, we assure that the suspension can, once it's basically dosed once a day, and the next morning you shake it, and then you dose it again. So you need to make sure it's resuspendable, that the dose uniformity is maintained throughout the in-use period. And we do something called in-use stability testing, where we actually are opening the bottle, setting it out, if it's a refrigerated product, for an hour each day for the duration of that in-use period, to make sure that there are no changes in the physical and chemical stability of the product. Likewise, one needs to conduct antimicrobial effectiveness testing. This is for a multi-use product, to make sure at time 0 the level of your antimicrobial agents is sufficient, and that at the end of shelf life, so that you still have success with that test, because during that full year in-use period, say 2 weeks into in-use, there are constant charges of objectionable organisms to the suspension, and you want to make sure over the course of the in-use period that you actually have it preserved in the proper way. And also, finally, photostability issues were conducted according to the ICH guidelines for this as well. So I wanted to give you an example of some of the results that we had generated for the lisinopril product. It's a 1 mg per ml suspension. The storage condition for the product, once it's constituted, is controlled room temperature. And we followed what we did to support putting this in the label. We actually looked at 3 unique tablet lots. Each of those lots reflect 3 different bulk drug lots, again, to show the robustness of the process across multiple manufacturing lots. We also had multiple scientists, if you will, follow the instructions and constitute the suspension to show that it was robust for different persons not familiar with the program to just follow the instructions and constitute the suspension to the right concentration to show robustness across bulk drug lots, as well as robustness across pharmacies preparing the preparation. And then we follow the stability. Stability is key for the active ingredient, but also for this product and its degradant, I will say. We also monitored the preservatives, again, with this particular formulation, with some of the changes that we had to make, especially monitoring the pH, et cetera. We did have to optimize the preservatives so we had adequate protection at the end of shelf life. So we actually followed the preservative levels also on stability with this product. And this is the in-use stability. So we have a single bottle. The bottles that were mentioned in the previous study were done by our standard techniques. That is, that you open the bottle and pull a stability sample. Each bottle is a unique stability point. And then we also had, in parallel, for the 3 different batches of API, we actually had a bottle that was the in-use sample, which was opened every day for an hour and then closed, and then we actually did stability time points across the in-use period, again, to make sure that the product was robust across multiple drug lots, and for the in-use period, the 4-week in-use period for development. And finally, again, for the 3 different lots, we actually looked at the stability in the presence of UV and visible light. Didn't see any issues, but again, for certain products, this does become very important, especially if they are light sensitive. But we did do that to provide that information in the label. So I did want to highlight antimicrobial effectiveness testing. I will say that the requirements for the U.S. and Europe are slightly different, so you need to make sure you understand those when you're conducting this testing. But for the lisinopril product, we were sailing along with the development until at the end of shelf life, the AME test came back as a failure. And the reason for that, again, was due to the calcium phosphate that was in the bulk drug tablet. What we ended up doing is modifying the formulation with a stronger buffer. And with the stronger buffer, we were able to overcome this titration with calcium phosphate and had success with the antimicrobial effectiveness tests. And I'm going to come back to this with the diligence aspect, because this worked great until ALSA decided to discontinue making the Bicitra product, which is something you need to monitor for these extemporaneous preparations. PK performance, I just put this here as a reference just to show that actually our clinical studies were actually conducted in adults and in pediatric patients with this extemporaneous preparation. And I know this is something that is not routinely available for extemporaneous preparations that may be done in pharmacies, but it is something that we had done as part of the development of these products. The other considerations have to do with acceptability, taste and smell, dosing volume, viscosity, mouthfeel, the measuring device and the precision of that device, as well as the ease of the preparation and administration and appearance. And I will say, for the preparations that I prepared, they did not require grinding with a mortar and pestle. In some of the original work that I had done, there were some concerns for the preparation pharmacist by doing that for some of our drugs. So you actually took the intact dosage form, put it in a small amount of water to disperse it, and then dilute it with the compounding vehicle. So there is no manipulation per se in the pharmacy, because I felt strongly that that was an important thing to consider, though I do know that there are some preparations out there where pharmacies are required to use a mortar and pestle to grind the dosage form, even in the presence of some of the vehicle beef before they make the suspension. Stability is interesting because there are levers that one can use in extemporaneous formulation development to actually impact the taste aspects of the dosage form. And this was particularly done for the losartan product. The actual drug in the dosage form is a potassium salt. It's very soluble, but not particularly tasty. And to overcome that, we simply titrated the drug with the compounding vehicle, and the resulting drug particle size, we actually did measure it, was a submicron particle size. We actually conduct disso testing because our dissolution was sensitive to the particle size of the bulk drug in this precipitated state. And as a consequence of this, again, I mentioned we had the sensitivity to various packages, and we actually had a 2 to 8 degree storage. If we stored this product at room temperature, we did start to see particle size growth over time. So again, we're monitoring the physical stability of this, but this particular change actually impacted the palatability of the resulting suspension. So what you actually see in the actual label is shown here. There's a lot of backstory behind the paragraph, I will say, and I just wanted to make sure that that was clear, that getting this simple paragraph that says, here's the suspension, you take the tablets, you put them in 10 mils of water, the bottle is specified, your shaking period, how long it has to stand, and how you dilute it with the subsequent vehicles, and the suspension itself, and then the stability and storage, all that information, the backstory that I presented sort of is all that's behind a simple paragraph that appears in the labeling for the product. So the other thing I will just mention briefly is diligence that's required in these pediatric products. And I will say that there are some examples many examples actually of extemporaneous preparations of product labels. And one I will mention briefly is the Tamiflu product. So Tamiflu does have a pediatric product available. It's the oral suspension. However, in the crisis of flu season, availability is not always present across all sites that are interested in this product, and so they actually include in their label for those purposes in emergency situations, as they state, the ability to compound the adult tablet to make the pediatric suspension. So I do think that there are some cases where these extemporaneous preparations, if they are really investigated and again provide the full backstory, can actually help in emergency situations to provide supply for patients, again, at that patient-doctor interface. I want the child to get this, the Tamiflu dose. It's not available. It's only available as the adult tablet. What can I do so that I can get it to the patient that's in need? And so I do think there's a place for these preparations in that vein. But they do require diligence. And what I mean by that is, for the lisinopril product that I've described, it had been on the market in the U.S. for about eight years before we received a notice that the manufacturer of the Bicitra product, and I will mention that this product was very important because it's a stronger buffer and it allowed us to overcome the calcium phosphate and therefore have successful AME testing at the beginning and end of shelf life for the product. So when they decided to remove that product from the market, there's a flurry of activity because now I don't have a product. If this is the preparation that I have available, I don't have this product. And so we had to identify suppliers of a similar material, and we had to work with our quality group to assess the regulatory status of these various vehicles and the excipients that they use. And then we had to source, after sourcing them and getting multiple lots to be representative of their process for evaluation, we then had to repeat the stability studies and more importantly, the antimicrobial effectiveness studies in order to make the change in the label to support this. And actually we had to do these studies and present this data to the regulatory bodies so that they could then update the label for the product. So once you have these extemporaneous formulations out there, you need to have diligence to monitor to make sure, one, there are no changes in any of the excipients or vehicles that you're using, and if there are changes, you need to assess those changes on the product so that it can be available and can have the same quality attributes as the products that you've developed. So what I want to do at this point is change gears a bit to something that's a bit more controversial. What I mean by this is that pediatric development now has, there's a great deal of development going on now, which is great. I will say, as a formulator, which I am, if I knew the pediatric doses before I started the development, getting the right potencies, et cetera, would be it's a more focused effort. However, when I start development, especially when we're doing this while the adult program, if you will, is still finishing up or starting their phase 3 studies, we might not know the dose. And actually, even though we may give a single dose for all adult patients, when we go into pediatric space routinely, we are dosing according to the age or weight of the patients. And if I go into younger and younger age cohorts, this challenge becomes greater and greater. So there's a requirement of dosing flexibility. That is one caveat. And the unknowns about what happens as you go to younger and younger age cohorts, it also makes it challenging from a formulation perspective. Some would say it makes it fun. I'm one of those people, but I'm just saying it's part of the journey. And the other thing that has happened more recently is that there is a desire from a regulatory perspective to simplify dosing tables, to minimize dosing errors, and make ease of administration greater for physicians and caregivers. So I need flexibility on one hand because there are unknowns, and on the other hand, I need to be simplified so that I can make sure that the formulation is designed to minimize those errors. And I want to give you a couple of examples of this because it really is one of the reasons why I think the discussion about extemporaneous formulations to assist in development could be very important. So little background on pediatric patients. If you go into newborns, it's like a new frontier. There is some data about the ontology of enzymes in newborns, but it's by no means complete. I know that there are additional studies ongoing about the GI contents, et cetera, of neonates relative to infants and toddlers, children and adolescents. So as you go to younger and younger age groups, you could end up with more and more unknowns. And from a development perspective, I'm trying to streamline the options, one, so that I have that simplified dosing table, and two, so that I actually have not created for my manufacturing colleagues a supply chain issue because the volumes are so low. Gastric emptying time can be impacted, pH, which I'll get to a slide on that next, the mobility of the intestine. These things are known to be different children going to infants, going to neonates. So the younger your age group, the more you end up into this area where we may not know as much about the environment in which the drug is going to be absorbed. I know that there's research going on with that, but while there are these unknowns, I think it does set up the formulation scientists for trying to get flexibility and not knowing exactly how much flexibility you're going to need. Gastric pHs is particularly interesting. It starts out high, then about two to 3 days into life, it drops down to being like an adult, and it jumps back up again and starts to progress back to being like an adult as the child is over a year in age. So if I have pH-sensitive release, or if the PK of my drug is impacted in this region, again, it's not a surprise, but you will have surprising effects on dose and PK as a consequence of these conditions, depending on the API. So I want to give you an example of this thought process. And again, this is not conventional thinking. It's just something that I think perhaps could be helpful. And again, this is only true in the U.S. Routinely, in the past, there have been 2 concentrations of Tylenol. And I believe that the 2 concentrations were designed so that we could minimize the actual volume of liquid that younger infants received. And so to minimize that volume, they actually had a greater concentration of drug in infant Tylenol versus children's Tylenol. And the packaging is clearly differentiated. However, routinely, physicians would write scripts and say, okay, give Johnny, give her 2 mils of Tylenol without necessarily specifying children's versus infant. And there were cases in the U.S. where children were overdosed because parents didn't realize that the 2 were a different concentration and gave the infant Tylenol to older children thinking that it was for infants, so it would be okay, and the children actually were overdosed with this compound. So the FDA actually drove the reformulation of Tylenol to a single concentration. Again, the drive here is to simplify the dosing table. There's 1 concentration, and the volumes are set according to that concentration. There's no A or B confusion. Is it infant? Is it children's? And it actually was done to minimize the errors of dosing that were seen in the marketplace. And I want to use this example and the second example for a program that we were developing to argue that if I went into my PK studies with a concentration, with a formulation, an extemporaneous formulation, and determine the PK of the drug in the target pediatric population, and then with that information would then better design my formulations to be the right dose or potency, or whatever potency flexibility that I need, I actually could design my formulations with simplification of the dosing table in mind. And the challenge is, I don't know before I start PK studies or get the PK data in pediatric patients across the age group, if I'm going to have to require a great deal of flexibility, or if it can be met with a single dose, if you will. And I think that you would see here with Tylenol, there are multiple doses, but by streamlining it to a single concentration to provide those multiple doses, you simplify the dosing table, minimize errors. And I think that is the new objective that is driving some of the regulatory questions around pediatric development. And it's something that's difficult to do without knowing how flexible one needs to be. So I want to give you an example of the raltegravir pediatric case. We started this development many years ago. The product has been approved for pediatric, which is great. We made some decisions about the dosage form, and because we didn't really have a bridging strategy, just to be honest, the formulation that we use for PK studies is the formulation that we also used for our safety and efficacy trials, and the formulation that's also on the marketplace, just being honest. So originally we developed 3 chewable tablet potencies for kids 2 years and older. The 100 mg and the 50 mg were weight multiples. That's pretty simple. The 25 mg was bracketed by the 100 and the 50, if you will, but it was, the PK is actually linear in this region, so it's interesting to look at that. And then we developed a powder for suspension for kids under the age of 2. It's a 100 milligram sachet. You make a 5 mg per mil suspension, and you adjust the dose with the syringe. And so you have one presentation that gives you flexibility for all the kids under the age of 2. And the challenge here was that we were going into neonates, and the dosing with neonates was, when we finally got there, was quite interesting, and I will get to that. But with each week of life for a neonate, from the time of birth to 4 weeks, each week of life with the maturation of the enzyme required a dose adjustment. So the original dosing table for the chewable tablets and for the suspension is noted here. So the chewable tablets, again, went to kids 2 and older, and the younger kids only got the powder for suspension where they were dosed on a mg per kg basis. The doses are outlined there. And we had run our PK studies, and we actually ran our safety and efficacy trials this way. And when we went to put our filing together, WHO was reviewing the data, and it was clear for them, instead of having all these different dosing bands, that if you look at their weight bands, you could actually provide the dose for kids 3 kilos, that is 2 years and older, just by using the 25 milligram tablet and multiples of it, or the 100 milligram tablet. So it's a simplification of the dosing table once you have that PK experience. And that proposal did open some eyes, because, you know, when I did speak with the clinical group about the 20 mg versus 30 milligram dose and what WHO is proposing to distill into the 25 milligram dose, for example, and are we overly cautious in dose, even though we do have tremendous safety margins with this molecule. But it's a case where, had I known this, I could have done my chewable tablet 25 mg increments, and that would have been for kids 2 and over. And it gets even more eye-opening. So again, the suspension for children from the time of birth to 4 weeks of age, with every week of age, there's an adjustment in the dose. And we provide that with that same powder for suspension. Looking at the dosing volumes here, which is particularly challenging, because we set that potency, you know, when we are running the kids that are 2 and under, and actually took us a while to get to neonates, we had to modify the concentration for the suspension so that we could increase the dosing volume somewhat, so that patients could, or caregivers could easily measure this with a dosing syringe. But one consideration is that we could develop a 5 milligram chewable tablet with a score. And that chewable tablet could be dispersed in formula or breast milk or water in like a teaspoon, and dose these younger children. So you could actually get, with a 5 milligram potency with a score, you could you could get the same dosing flexibility that's provided here, but the presentation to the caregiver is much simpler. So the question when I look at this is, could extemporaneous preparations in the course of pediatric development help us to develop formulations and potency options with simplification and ease of administration in mind? And right now, I will be honest, we don't do this. We tend to go into our pediatric PK studies with the formulations that we think will give us adequate flexibility, and we'll work out the dose table as we go. Though I think there is it would be a great thing if we could, using that PK information in a small number of pediatric patients, and again, working with our modeling and simulation teams and our clinical teams to look at the dosing table before we go to safety and efficacy with the thought in mind of how is the caregiver going to deliver the medicine to the child. So with that, I'll just end by saying the development of formulations is a partnership. Tools to streamline development can actually help not only the formulators, but help the caregivers in delivering the optimal formulations and formulation solutions for pediatric patients. And while I know that right now there is not a huge push to do PK studies with, if you will, a fit-for-purpose approach for pediatrics, optimizing it once you know the PK and what the doses are, to actually rolling into your formulation development, I do think it's something that we should consider because it allows us to provide the formulation meeting all of the requirements that the regulatory bodies have. That is, a formulation that is robust, that provides us the PK information, that also is designed with the caregiver and the environment in which this is going to be dosed in mind. And with that, thank you for your attention. I do thank the lisinopril, losartan, and raltegravir pediatric development teams. I've learned many lessons in the course of the development of these various products, and I open this up for any questions.
Okay. At this time, I have a question from Yogesh Patel from Amgen. In lisinopril suspension drug product, was there any concern for using parabens as preservatives from regulatory agencies?
There were questions. We had to show data that with the parabens present, we actually had successful antimicrobial activity at lower overall levels of preservative in the formulation. And what I mean by that is, without the parabens, the potassium sorbate that's also present in the formulation would need to be at a higher concentration to provide adequate protection, because the parabens are exquisite at killing mold. I'm just being honest. So they do serve a purpose, and you do have to engage in a discussion of the risk-benefit of that. But there is by putting those into the formulation, the overall preservative load was lower for the formulation.
Okay, thank you. The next question is from Wen Chin Fu. The majority of extemporaneous preparations are still off-label. These have minimal stability data and no bioavailability PK data. What is your opinion on this?
So in the course of doing research for this talk, I will be honest, I saw some extemporaneous formulations of products that I had worked on. And the thing that I will say is that I understand the liabilities of the compound because I'm part of the development of the product. And a person coming in saying, oh, I will just take the contents of this capsule and I'll grind it with a mortar and pestle and then I can do these things. They're doing that without necessarily understanding what's in that composition and what the liabilities are with that particular API. And so I was kind of cringing when I read some of these recipes because it's clear that they don't know, for example, that this is a nanodispersion. So the real issue is not chemical stability at all. It'll show up as being very chemically stable, but if the particle size grows beyond that nanocrystal, you have a PK impact. I don't think that there's an appreciation of that necessarily for a person who's designing this recipe without that information. So I do think that by sharing some of these, I think that we overall impact our patients and physicians. They know what they're going to get because they have that understanding. I don't think necessarily because some of this information may not be out there, I don't think that pharmacies compounding things actually have that appreciation. I'm just being honest.
Okay, thanks, Karen. I have a question from Lorraine Sequier. Thanks for the very interesting talk. Did you receive any challenges from regulators regarding the use of preservatives in your formulation for pediatrics? If yes, how did you overcome these challenges?
So we did receive challenges, as I mentioned, and we actually shared with them data of the result of we actually had to go through with the manufacturer. Actually, the vendor made us totally non-preserved vehicle, and then we had to show, basically justify the level of preservative that was in there, and when we showed them those results, they were okay with the formulation. But we did have to provide that data.
Okay. Hi, thanks. I have a question from Ray Z. Do you usually perform dosing accuracy study on using actual dosing syringe or cup? When doing in-use stability, do you use dosing syringe to sample stability samples?
So we actually do dose accuracy studies with our syringes. I will say these are different from human factor studies, I'm just going to be honest, but we do dose accuracy study for the syringes. We also, when with taking our samples for stability, the in-use portion, we have used syringes just to make sure we can use that syringe over the in-use period of 30 days or whatever it is, to make sure that, if you're cleaning it and then reusing it, that it's still working. And we have worked with some syringe suppliers to generate that data. So yeah, we do run those studies.
Okay, thank you. I have a question from Anthony Nunn. How do you assure the quality of commercial diluents or suspending agents when the quantitative data is not made available, nor is information made available to the standard expected of APIs and excipients, at least in Europe?
So what we did when we put this product in our actual label, we actually had our quality group work with that vendor, and actually we saw their manufacturing process and we looked at their excipients and made sure that they met our standards. And then we actually signed an agreement with the manufacturer, again, so that they would notify us of any changes, either in excipient grades or manufacturing process, so we could assess that for the product. So there is a relationship that you need to build with your vehicle supplier, very much like the relationship that you have with an excipient supplier. I'll just be honest.
Okay, thank you. I have a question from Madhavi Srikoti, Bristol Myers Squibb. Were there any regulatory questions on DKP amount?
For the lisinopril product, no, but we kept those levels within the specs that we have for the adult product, and those levels had been qualified in our preclinical safety studies and in juvenile animal tox studies as well. So we had the data to justify those levels.
Okay, so you had those on specification. Okay.
Yes.
Okay. Another question from Lorraine Sequier. How did you assess the palatability of your formulation?
That's very good. I will be honest, for the losartan product, before we went into pediatric patients, because when I was developing this product, our marketing colleagues were very much concerned about the dose, and it was a balancing act, so we actually had our adults basically tasting this product to see if it was acceptable. Then we also, during the course of our studies, had questionnaires about acceptability for the caregivers during the course of the study, and also monitored the ability of our patients to adhere to the dosing regimen, because this is those particular drugs you would take these chronically in pediatric patients. So we used all of that data to support the acceptable taste of the formulation.
Okay, thanks. And one more question from Anthony Nunn. This is the last question I have right now. Okay, I'm sorry. This is a follow-up to his last question regarding assuring the quality of commercial diluent suspending agents. But for individual pharmacists making the product, did you want me to go further on the previous question?
So for individual pharmacists making the product, part of the reason why we had done, that preparation, multiple people preparing it, is to make sure the instructions were easy to follow. We also conducted our clinical trials with this extemporaneous preparation again. So we had pharmacies across participating in our clinical studies, actually preparing this daily for caregivers. And we actually, when we had our investigator meetings, we stepped through that preparation to make sure there were no questions in terms of the quality of and we specify clearly what vehicles they were to use. In some cases, I will be honest, because of the availability of supply, in some cases for our clinical studies, we actually ship material to them. But for the products that were available, they were specified clearly, and then the instructions, when we had our investigator meetings to make sure that the pharmacists could clearly follow our instructions, we did not have any issues with that.
Okay. Thanks, Karen. And at this time, I'd like to conclude the questions. I have no further questions. Actually, I'm sorry. I guess we have one more minute for one last question here that just popped up. You mentioned dissolution testing and particle size affecting dissolution. Is the dissolution test for suspension part of product release specification?
Yes, it is.
Okay.
Yes, it is.
All right. Thank you. Okay, so I would like to thank you very much, Karen, for a very interesting and informative talk. At this time, there's no further questions. I'd like to note that at the conclusion of this webinar, everyone will receive an email from Mahalea with a feedback survey. And it's very important to Karen as well as to us as moderators to ensure the quality and the value of this webinar series. So we will appreciate your feedback. And please join us for our next webinar on November 2nd on intermediate release reverse enteric-coated melt spray congeal microspheres as a pediatric platform. So again, thank you very much, Karen, and I will turn it back over to Mahalea.
Thanks, Beth. Thanks, Karen, very much again for your really informative and interesting talk today. This will conclude the webinar. As Beth mentioned, you will receive a feedback survey, which is incredibly valuable to both the working group, the speaker, and to IQ as a whole. Please do take just a moment to respond to those questions and join us for the following webinar to take place later this year. And you will receive also a link to this recording, and it will also be available on the IQ website and for IQ members on IQ Connect. Thanks very much, and I hope you all have a great rest of the week and a great weekend. Thank you.